Hello viewer, today I will talk about different shape and size of the protein as well as the structure and function of the fibrous protein. I will talk about the primary structure of insulin, tertiary structure and function of fibrous protein like collagen, keratin, elastin, fibroin as well as actin. I will talk about the quaternary structure of the globular protein carboxypeptidase A as well as tobacco mosaic virus coat protein, the topology of the membrane protein as well as I will talk about the technique, database, disorder related to the protein structure. As we know that proteins have different function as well as protein have different size and shape. Round structure is found in case of hemoglobin and myoglobin, long structure collagen. We can find the elastic structure in titin as well as we can find strong structure of spectrin. Now, when we are talking about the characteristics, the shape and the size of the protein is dictated by the amino acid sequence. The amino acid sequence, hydrophobic sequence dictates how the protein will be folded. Protein will be folded in the stable single conformation by release of small amount of energy. So, it is marginally stable. And in case of multi-domain protein, we can find different independent subunit, larger molecular weight protein. Structure depiction, alpha, beta, so these all structures have different propensities to fold. They make crystalline array of the protein. The membrane protein have hexagonal array. As we know that the protein family serine protease like trypsin, chymotrypsin, so there is a small signature sequence, so which actually relates the homologies of the protein, close relative according to the structure as well as function. Domain shuffling of the block, that is protein sequence or structure according to the evolution can also be possible. Structure depiction, as we know that the extracellular protein are the fibrous, collagen-like. So, disulfide linkages are present as well as we know that the head-to-head or head-to-tail arrangement is there. Head-to-tail arrangement is present in neuraminidase. Protein can be assembled, disassembled. Now, for these assemblies, sometimes independent folding can be happened either, or it may need the help of accessory factors like protein disulfide isomerase, cis-transprolyl isomerase as well as the presence of chaperonin protein like GROEL, GROES, HSP, heat shock protein 60, 70, etc. Now, why the large subunit is needed? Just for the genetic information, the proper assembly, disassembly can also be controlled. Next one, primary structure of the insulin. So, in case of insulin, first it is synthesized in pre-proinsulin and that exists within the cell, in every cell. Now, by pre-proinsulin, protease remove the C-peptide which circulates in the circulation and it makes two chains, A chain, B chain. A chain is called as glycine chain and B chain is phenylalanine chain, and they are interconnected by interchain disulfide linkage in between 7 cysteine residues of both A and B chain as well as 20th residue of A chain cysteine and 19th residue of the B chain cysteine. And this can be made by the recombinant DNA technology nowadays. The N-terminus is getting changed from threonine to alanine in case of porcine to the human. As we know that, so after getting synthesis, it is getting secreted as pre-proinsulin, but three-dimensional structure is getting folded by the action of the protease and proper protein folding. We know that the fibrous proteins are insoluble in the water and the isoleucine, valine, these residues make the inner core. So, they have the rope-like structure. Not only that, the repetitive sequences are present and these repetitive sequences make it flexible, extended structure as well as some supple structure, flexible structure is possible for the silk fibroin, etc. As we know that the microcrystalline structure, microscopic structure can be observed in case of fibrous protein, but collagen gives the strength as well as elastin gives the elasticity, rubbery network. Fibroglobular protein. In case of some animal and plant, some proteins are present. In case of G-actin, it is globular, but when they polymerize, they make the F-actin fibrous protein. Alpha beta tubulin, they make microtubule, that is fibrous in nature. So that is called the pseudoglobular protein. So they are not truly globular or truly fibrous. Globular protein make the fibrous structure. Next, comparison in between fibrous and globular protein. Globular protein have compact structure, compact fold, but in case of fibrous protein, it is extended structure. Globular protein are soluble in water, but in case of fibrous protein, that is insoluble in water. In case of globular protein, different types of secondary bonds are present. Different types of secondary bond means we can find alpha, beta, they are actually getting hold together, different structure, secondary structure are present. But in case of fibrous protein, only one secondary structure is present. Quaternary structure are held by the covalent forces, but the fibrous protein are held by the covalent bridges. This globular protein can be engaged in several cellular metabolic function as enzymes, transport through membrane, metabolic regulation, as well as messenger through proper membrane. But fibrous protein, they are needed for the structural role, bone, skin, tendon, blood vessel, etc. In case of fibrous protein, we can find the secondary bonds are involved to make the cross link, to connect the interchains. Fibrous protein type. I will talk about collagen, alpha and beta keratin, silk fibroin, elastin and actin filament. Collagen structure. So collagen structure is mainly made by the tropocollagen unit made by the alpha polypeptide chain, and 3 helical alpha peptide, they actually intertwine to give the main tropocollagen unit, which is vertically arranged to give the tropocollagen structure, and these tropocollagen make the collagen fibril structure. Now this tropocollagen has 300 nanometer length and 1.5 nanometer width. We can see the presence of glyxy to the power n, that the repetition of this residue in collagen. Why? Because for every intertwine of the alpha polypeptide chain in the triple helix tropocollagen structure, internal residue glycine. So glycine actually inhibit the steric hindrance, and proline actually give the flexibility, and X and Y are the proline and hydroxyproline. This hydroxyproline is actually giving the interchain hydrogen bonding. So proline by post-translational modification makes 4-hydroxyproline by the help of prolyl hydroxylase. It needs the help of vitamin C as coenzyme or cofactor. So in the deficiency of the vitamin C, the scurvy occurs. Scurvy is malformation of the collagen structure, muscular dystrophy, fragility, so bleeding gums can be happen, we can find here. Not only that, so there are so many interconnection in between 3 alpha chains, so that they actually make the triple helix of the tropocollagen. So what are they? First, with the help of lysyl oxidase, the lysine is getting converted to the L-lysine. Now 2 L-lysine of the 2 alpha chain will make the aldol condensation, L-lysine aldol. Now this L-lysine aldol along with the histidine of other chain make the Schiff base intermediate aldol histidine. Now these aldol histidine along with the different chain 5-hydroxylysine make the merodesmosine derivative by bound with hydrogen bonding. So in this way, secondary amide bond, Schiff base, aldol condensation help to connect all 3 alpha chains to make the tropocollagen structure. So the biochemical importance includes that I have already told about the scurvy. Not only that, it also includes another disease, Menkes disease, where it reduces the copper absorption by the intestinal lumen. So the copper deficiency causes the reduction in the efficiency of lysyl oxidase, which need copper as the cofactor. So it causes the muscular dystrophy, fragility of the collagen structure. Not only that, lathyrism is also found by beta aminopropionitrile generated from Lathyrus odoratus, Khesari dal. So this is beta-aminopropionitrile is the inhibitor of lysyl oxidase. Now what I have told that these alpha chain, we can find the pitch is 1 nanometer and 3.3 residues per turn or coil. And when they will intertwine with each other, we can find the 8.6 nanometer of the pitch and 10 gly-XY residues per coil turn. And we can see that every chain is alpha chain is twisted in the left-handed manner, but when the triple helical coil is twisted, that is in the right-handed coil. So when we are talking about the collagen function, we can see in the opposite direction twist, it actually raises the tension, and we can find this gives the strength in the cartilage, bone, etc. Blood vessel. According to source, type 1 to type 4 collagen are present. It makes the skin, it makes the cartilage, tendon, bone, blood vessel, etc. And we can find in case of collagen fibril structure, 67 nanometer is length and 10 to 300 nanometer breadth. Gelatin that have no food value that can be derived from collagen. Keratin structure. So alpha keratin and beta keratin. In case of alpha keratin, we can find the amino and carboxy terminal are at the same site, and we can find the coiled coil dimer. So they are getting dimerized to give the protofilament or profibril structure in the N to C terminal end. Now this protofilament profibril makes the microfibril structure. Microfibril make the macrofibril structure. Macrofibril make the supercoil structure of the hair. We can find 300 hydrophobic residues at 4th and 7th amino acid sequence that is very much insoluble in water, and they are connected by interchain disulfide linkage and hydrogen bonding. Now interestingly, the source of keratin function is actually found in hair, nail, wool, etc. Now this disulfide linkage can be reoxidized, reduced by help of oxidizing agent. So permanent wave of the hair, perming, curling, that is also possible by changing the interchain disulfide linkage. Now when these A, B, C, D, E, F, G, so this heptameric sequence is actually repetitive, and A and D are the non-polar residues. We can find there are several non-polar residues are there, and in case of hair, if this particular alpha keratin is not well formed, that causes the disease epidermolysis bullosa simplex. Beta keratin. Beta keratin have beta strand structure, and this beta keratin according to structure and function is found over the reptilian skin, and we can find it is stratum corneum of the reptilia. We can find their scale over there in the beaks we can also find, and interestingly they actually act as waterproof. They prove the desiccation. Silk fibroin structure, beta strand of zigzag structure. So parallelly anti-parallel beta strands are actually oriented. We can find the glycate structure, and here actually this repetition is intermediate, it interrupted by the tyrosine residues. These actually make the microcrystalline structure. Now silk fibroin, so we can find the alanine, glycine, valine parallelly oriented structure to make the spider web cocoon. So this is the supple structure where actually they are make the different strands of beta strand, they are actually intersliding over each other. Elastin structure. Elastin is actually present along with the collagen. Collagen gives the strength, but elastin actually the flexible structure, extended structure. It is also made with gly, ala, val, etc. But lysine and proline make the desmosine cross-linking. But actually we can find the cross-linking inhibit the extension up to some limit, so that after snapback, after removing the tension, it will go to the snapback condition. Not only that, this elastin structure is getting changed, the rubbery network, the cross-linking are in between the 4 structure, 4 helices, 4 interchain. Now, these cross-linking actually correlate the interchange between fibril and rubbery network of the elastin. Elastin made by the tropoelastin and found desmosine and merodesmosine, isodesmosine, etc. Now, elastin is present within the tendon along with the collagen structure, and it gives the flexibility, extendability, etc. So, it is present tendon cartilage at that particular region. Actin filament. So, actin filament is made by the two types of filament, that is microfilament and thin filament. Now, G filament or G actin is globular in structure. They make the fibrous F actin. They have the ATPase action. So, they actually make the microtubule structure that is needed for the cell division, to make cellular network, framework. It is needed also for the motility of the cell. So, actin is very important fibril structure. Quaternary structure, globular structure of carboxypeptidase A. So, this is pancreatic exopeptidase. It is sometimes called as CPA1 or CPA2. We can find different domain structures here. These CPA1 and CPA2, 8 alpha helices and beta strands are present, and interchain hydrogen and disulfide linkages are also present. So, different domain structure, globular structure is also present here. Tobacco mosaic virus. So, coat protein is actually the cylindrical. So, they protect the RNA, the tubular structure. It gives the protein-protein interaction. It is 158 amino acid residue containing protein. Apart from these, rhinovirus have 64 subunit containing protein, make the spherical shell of the protein together. Membrane protein. Membrane protein may be extrinsic, peripheral, can be removed just by sodium chloride concentration, or may be intrinsic. Intrinsic may be covalently attached intrinsic monotopic protein or integral monotopic protein as GPI anchored protein, mid-stylation, etc. Or may be the transmembrane protein totally passed through the plasma membrane, that is permanently attached to the membrane. This transmembrane protein can be made by the alpha helix or beta sheet. Porine. Porine is made totally the beta barrel structure, beta strands. It is usually the trimeric protein, and trimeric each monomer made by the 16 beta strands. Here the inside out model can be observed. So, the outer residues are made by the hydrophobic amino acids which can interact with the fatty acyl chain of the membrane, but the inner core is made by the hydrophilic amino acid to make the hydrophilic channel for the passage of the membrane. So, porine structure made by the beta strands. Bacteriorhodopsin, so it acts as the proton pump. It is needed in case of halobacteria and archaea. The 7 membrane spanning protein. It is harvesting light energy and convert it to the chemical energy, and after receiving energy 120-degree rotation possible. So, it spans the membrane 7 times, made by the alpha helices, bacteriorhodopsin. Hydropathy plot. So, this is the plot that how the hydrophobicity control the native conformation to fold. We can follow the axis of the hydropathy index with respect to the hydrophobic amino acid residues. So, there are the propensities, the predictions, how the protein will be fold, which structures, which amino acids will be out, which will be inside, regarding the topology of the membrane. Because here we can follow the presence of 7 spanning bacteriorhodopsin, 7 patches of hydrophobic residues are there, along with single spanning glycophorin. So, peaks are there indicating the energy needed for the hydrophobic interaction, that is called as hydropathy index. It is specially to determine the topology of the transmembrane protein. Protein denaturation and the renaturation. We know that protein can be destroyed by application of the heat. 3-dimensional structure will be destroyed to make the linear sequence of the protein. But if by 40 or 50 degrees centigrade, if total peptide structure is not destroyed, so after application of heat or 8 molar urea, if they can be withdrawn, so disulfide bond will help to renature them off. Otherwise, they will be destroyed permanently. Hofmeister series. It is the series of the ions needed for the salting in, salting out, solubility as well as destruction of the protein. As we know that the earlier member are helpful for the salt out and the later member are helpful for salt in. If it destabilizes the protein structure like thiocyanate SCN minus, so they are called as chaotropic ions, or cosmotropic ion help to solubilize them of make the proper folding, stabilizes the folding structure. Techniques to determine the protein structure, so many techniques are there like FRET technique, CD spectra, NMR spectra, EPR electron paramagnetic resonance. Different techniques are responsible to determine the protein structure. Now we know that disease related to the malformation of the protein structure. So some protein can be misfolded, okay, like amyloid fibril in case of Alzheimer's disease, Parkinson disease. So they cause the neurodegenerative diseases or neurodegeneration. Protein structural analysis can be done by the X-ray crystallography as well as NMR study. So X-ray crystallography gives the atomic resolution. So native conformation give how the biological function can be shown. In case of NMR study, simpler version of the protein structure can be known. So the external residues R chain varies according to the polarity but having relaxed conformation. The hydrophobic residue make the core structure. So that's all aspect can be described by the NMR study. Protein structural bioinformatics. So the protein database, the protein structure can be known by the specific site, that is http://www.rcsb.org/pdb protein data bank. We know the protein modeling by means of ncbi.nlm.nih.gov. Protein structural classification can be known by the SCOP, SCOP, as well as we know it from CATH, CATH, VAST. Different types of software, different types of bioinformatic databases are there. Now, interesting thing is that protein evolution. In case of cytochrome oxidase, the cysteine, lysine, glycine conserved sequences are there. So genetic duplication, actually convergent, divergent evolution maintains the protein structure that can be known by the database. As well as these protein is related to the folding, BPTI protein fold in the ordered manner, amino acid sequence, they dictate how to fold. And we know that protein has nucleation condensation model as well as diffusion collision model to fold. And we know these protein folding, the post-translational modification sometimes inhibit protein proper folding. Protein structural prediction can be done by Chow-Fasman scheme. So alpha helix have propensity to fold in a specific manner, and this reverse turn minimizes the hydrophobicity, and different alpha helical beta sheet structure can be folded. Not only that, protein dynamics. These protein dynamics actually include the atomic fluctuation, the cohesive motion, as well as we know the triggered conformational change. And these sort of conformation change, the internal residue change, core structure change can be monitored via pulsed H/D exchange. The infrequent motion can be obtained via H exchange, hydrogen exchange, whatever, and atomic flipping give the idea about the different motion in the protein. I have already described about different size, shape of the protein as well as structure and function of the fibrous protein. In case of main protein, we know that the globular protein, that scleroprotein is present, as well as scleroprotein is the fibrous protein and membrane protein. Three distinct classes are there: globular, fibrous as well as membrane protein. In this lecture, I have discussed about the aspects of the fibrous protein as well as topology of the membrane protein. Thank you.